Cloning 101 with Dr. Jim Pickens. Cloning is the process of extracting DNA from a donor organism in order to form a copy of that organism. The DNA is taken from the donor cell and then injected into the new embryo cell, and then that cell is put inside the womb of the host animal until it grows into a organism that looks exactly like, physically exact copy of the donor organism. Contrary to popular belief, not all clones look exactly identical. Calico cats, for example, have two X chromosomes that differ randomly, causing the fur to be different colors no matter how exact you try to clone them. Take these bees, for example. One of them is much brighter colored than the other one. Their chromosomes differ when they were cloned, causing their colors to change randomly. We first began cloning cells in the 1970s when researchers in the lab took mice embryos and split them into test tube, and then put the resulting embryos in adult female mouse wombs. It wasn't until 1996 when we finally cloned a sheep named Dolly from a sheep's udder. She was the first successful mammal to be cloned after 207 tries. Oh, I'm sorry, that's 276 attempts. 2 years after Dolly was cloned, scientists in Japan cloned 8 calves from one cow. Sadly, only 4 survived. Another example of cloning is De-Extinction. That's where they take the DNA of an extinct animal and they use it to bring it back from extinction, such as the bucardo, a relative of the ibex. And one day, researchers hope to bring back animals such as the Tasmanian tiger, the woolly mammoth, and others, so we can hopefully turn this into this. The problems with reproductive cloning is it's very expensive, it can take a long time, and it has a low success rate. The major problems during this process are the psychological effects, genetic damage, and the damage to the mothers during the implantation and birthing process. This has been Cloning 101 with Dr. Jim Pickens. And now it's time for cloning the Jim Pickens way. Now this is why we don't clone humans. See, this Neanderthal bit this turkey's skull wide open. Now, we believe it had some sort of genetic mutation that caused it to go berserk. It attacked everyone in the facility. We had to put it down, and this is all that remains. Pickens, and I'd like to introduce you to where we store our DNA. Now, if you take a look right here, we have all the animal DNA. That one's probably from some sort of fish or something. Oh, there's the fish DNA, and then we have all kinds of dinosaurs. Now, we did get these from Jurassic Park, so you know, there's a mastodon, there's a bunch of velociraptors, and there's a T-Rex. So, yeah. Oh, and that you weren't supposed to see that. That's our human DNA storage right there. Now, in the process of cloning, the first step you want to do is you want to take a donor cell from a living organism. Such as this cell here. Now, after you've taken this donor cell, you want to take your host organism. Now, you put the donor cell into an embryo. You put the DNA into a donor cell embryo cell, and then you take that and you stick it inside the host animal. And now, after 6 months later, you get we're experiencing technical difficulties. Now that we've injected the DNA into an egg, we put it inside the incubator. Now we put it in the incubator for 10 seconds. Oh, it's done. Voila!
fully grown turkey.
If you have any questions, please feel free to ask my associate.